Hello everyone, welcome to today's live broadcast, Big Data and the Learning Healthcare System, Practical Applications for the Clinical Laboratory. I'm Robert Castellanos of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. This is an educational webinar and thus also free continuing education credits. After the webinar is over, please click on the C button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I'd like to introduce today's speaker, Dr. Lee Schroeder. Dr. Schroeder is currently assistant professor at the University of Michigan, where he is director of point-of-care testing and associate director of chemical pathology. His academic focus is at the interface of clinical informatics and health services research, using decision analytic approaches to model and improve the impact of laboratory medicine. This has included research involving laboratory capacity and quality improvement of sub-Saharan African laboratories, modeling the impact of different testing algorithms on health outcomes, and data mining the electronic medical record to better understand the performance of the point-of-care testing. Lee Schroeder completed his BA in physics at the University of Pennsylvania in 1995. His MD PhD with the NIH Medical Scientist Training Program Scholarship Award at the University of Southern California, San Diego in 2011, and his pathology residency, Stanford University in 2014. Lee currently serves as co-director of a Blue Cross Blue Shield funded pilot program to improve utilization of genetic testing for hereditary cancer syndromes and is a member of the Chemistry Resource Committee at the College of American Pathologists. I'll now turn it over to the doctor for his presentation.
So I'm very excited to be presenting on this topic, and I think because I feel like this is, in some sense, the Wild West of healthcare delivery. And the basic questions are still being asked of what can be done with big data and the learning healthcare system. And we've seen what big data has done in business analytics and web analytics, and this is going to happen in healthcare. It's a matter of time. So I think we're all familiar with what this cartoon is trying to convey. A lot of effort has gone into putting data into electronic health record, and it's really useful, obviously, for very many reasons, but it's not quite as useful as one would hope. You can hop on Google, search the web, but it's really hard to get interesting data out of the health record. And so that's what we're talking about today. And the hope is that electronic health records will make it possible to develop institution-specific tools and metrics to improve and measure patient outcomes. And I guess this is as good of a time as any to talk about what big data is. And of course, it's large in size, and sometimes it's defined as data sets that are a little too large or maybe way too large to use traditional tools like spreadsheets. But it's also typically like messy data that is what used to be thrown out, right? But maybe you can do something with this. And, so it's not an RCT, but there's still information there. And I think also it's defined somewhat in the questions you ask, that you get big enough data sets, you can start asking new and interesting questions. Questions like predicting readmission rate or chance of readmission of a patient, or maybe trying to glean off-label use of medication by sifting through the medical records. So the idea of using big data to improve healthcare is symbolized in the learning healthcare system. And so this actually started, I think, with the Institute of Medicine Roundtable on Evidence-Based Medicine in 2007. One of their quotes is that care that is important is often not delivered, and care that is delivered is often not important. And so, this needs to change. And, one of the common statistics is that it takes 17 years before positive research results are implemented into clinical practice on average and start improving patient care. That's simply not good enough. And evidence-based medicine has been based on the randomized clinical trial, and of course it will continue to be. But the problems with the RCTs are that they're expensive. There's narrow inclusion criteria, limiting generalizability of the study findings and personalized care. And in the end, not a whole lot of guidelines are based on RCTs. So this is a 2009 study, but things probably haven't changed too much. But even in cardiology, only 19% of the guidelines were based on randomized trials. So the learning healthcare system, I think, can be summed up by these two statements. One is to actively learn from the vast array of routine patient management decisions occurring every day in the hospital, so don't throw it away. And also to use the EMR as a decision support tool to personalize medicine to your population. So I did my training at Stanford in pathology, and there was this excellent example of a learning healthcare system there, which was published. But I started with a 13-year-old patient with lupus, admitted with nephrotic range proteinuria, antiphospholipid antibodies, and pancreatitis. And although anticoagulation is not standard practice for children with lupus, even when they're critically ill, with these risk factors, was that enough to anticoagulate? And there was mixed opinion among the faculty. Well, the lead author on this paper and the provider and the patient turned out, it was an informaticist. And so she was facile with what Stanford had put together in their clinical data warehouse. So they had annotated the textual notes, the free text notes in their clinical data warehouse with biomedical terms, and actually a hierarchical ontology of biomedical terms, so you could easily search through the record. So this provider simply counted up how many patients were like hers in the medical record, and she found 98. And she found that 10 of them ended up getting thrombosis during their stay. And then she went through and counted up, well, how many of them looked just like my patient? So with having heavy proteinuria or antiphospholipid antibodies or pancreatitis, and simply counting up historically, she was able to calculate relative risks associated with each of these risk factors for the populations at Stanford. And with that data, they decided they clearly needed to anticoagulate, which they did. And what was interesting is all this work was done in less than 4 hours by a single clinician, which is really great. It's not much time, but it's a huge amount of time, right, for regular patient care. And so that's when actually Naeem Shah, he's involved in annotating the clinical data warehouse, but him and others published this influential op-ed in Health Affairs, and the idea was that there should be a button, right, a green button that a provider could use to aggregate the data in the health record, in electronic health records, with the push of a button, and immediately see sort of historically what a patient management decisions, how they resulted for patients like yours. Well, we're not there yet by any means, but there's certainly big data activity in healthcare. So different groups are using records to predict readmissions, which is important, of course, with pay for performance, and eventually capitated models. Predicting risk of decompensation, discovery biomarkers, looking at drug interactions or off-label use. The Pittsburgh Health Data Alliance, so this is a University of Pittsburgh Medical Center collaboration. They're incorporating medical and insurance records, wearable sensors, genetic data, and social media to predict and optimal management decisions. Apple and IBM have a recent collaboration to take iPhone and Apple Watch data and analyze it in Watson. And but we're talking about the clinical diagnostics right now, and so there's a recent partnership between Quest Diagnostics and Devo it's an analytics company to create decision support for physicians. So Quest is offering up 20 billion laboratory results, and they're going to try to identify patients who might benefit from therapies like the PCSK9 inhibitors instead of statins, or identify those with rare diseases who might benefit from orphan drugs. So that's exciting. Now, the tools you're going to need, I think we need to comment on this because the definition of big data in some sense is not using traditional tools. And there's a couple open source options which are becoming the dominant options in healthcare, I think. So one is using R, statistical programming environment, and I highly recommend this. It's open source, it's free. There are packages constantly created by statisticians, so the latest biostatistical methods can be easily plugged in, and it's a very approachable language, scripting language. Python is a little bit more basic and a little more difficult to approach, but you can do, it's got a little more flexibility. And of course, then there's Excel and other commercial products like Tableau, which you can take spreadsheets and there's built-in analytics. I haven't had much experience with those. So what I want to take you through today is a few examples of mining the laboratory information system or electronic health record from the perspective of the clinical laboratory. And this can include reference range generation, looking at encoded data in routine lab panels, trying to evaluate accuracy of point-of-care instrumentation, looking at hemolysis, and doing some modeling to try to synthesize the information you find. So this is a great paper detailing the history of the reference range and reference intervals. And it hasn't always been the case that there are reference intervals attached to laboratory results. But now, even with the fanciest decision support tools and artificial intelligence, expert systems, probably the most important decision support tool in healthcare and most commonly used is the humble reference interval. And but there's some problems with it. So guidelines typically recommend 120 patients per subgroup you want to look at, and that's burdensome. You have to identify 120 healthy patients, and if you want to look at different age groups, that number can quickly blast them out of control. Now, what I want to talk about is auto-generating these reference ranges using data from the medical record. And why would that be useful? Well, probably the most immediate use would be for analytes in which there is no reference range published. And this is often the case in pediatrics, where the reference range is probably changing quite often as they grow older, and it's hard to get healthy pediatric samples, so there's just not a lot of reference ranges. And so that could be a really great low-hanging fruit for reference interval generation from EMR. Another thing is to normalize institution-specific instrumentation and population. So a lot of times one needs to just verify someone else's reference range for their application, and sometimes it's not even using the same instrument. So if you use your own data, then you're going to be using data from your electronic health record, you'll be using data from your own instrument and your own population. And we think now, and more with personalization of medicine, that different ethnicities, age groups, demographics will possibly have different reference ranges. That's the case with creatinine, and that's seen in calculations for estimated glomerular filtration rate. You have ethnicity in that equation. Well, we don't have different reference ranges typically for creatinine, but why not? So the data mining approach for reference interval calculation, there's two primary approaches. So one are indirect approaches. These are purely statistical, where, for instance, you take some central percentages. Maybe you plot out a cumulative or a CDF and look for the linear range of this cumulative distribution function, so basically removing outliers, and then take the central 95% of the population, and that's not consistent with International Federation of Clinical Chemistry or CLSI guidelines, which say you need to explicitly state which patients you're excluding. And in this statistical approach, you don't do that. So the other option is the a posteriori method, in which you explicitly identify exclusion criteria and then remove ICD-9 codes. Remove patients who have those ICD-9 codes. So here's a study of a posteriori method for hemoglobin, where an expert panel got together and identified a bunch of ICD-9 codes that should be excluded. It's a huge list. It's a lot of work. They probably missed some things, right? So it's not ideal, but it is an approach. Well, when I was at Stanford, so this is work done by Sarah Poole, who's grad student there in Nigam Shah's lab. And so I was involved in this project of automatically generating reference ranges from their Stride clinical data warehouse. And the idea is that, let's say you're looking at hemoglobin, so you query for your hemoglobin values, as well as all the ICD-9 codes for all the encounters for each patient. Then you identify outliers in the distribution of hemoglobin values and see if there are any enriched ICD-9 codes in those outliers. And if there are some ICD-9 codes that happen to be present more often in those outliers, well, you consider that an excluded ICD-9, remove all those patients with that code, and then rerun the script, the algorithm, and see if you still have outliers, and if you do, if there's any statistically significant enrichment of some ICD-9 codes. And you do this iteratively until you identify all the ICD-9 codes that are going to be enriched statistically. And now you've got your essentially automated a posteriori method. And then with this cleaned up group, you can take the central 95% as your reference interval. So we wanted to test this out on hemoglobin, which we knew was going to be tough because everyone knows most hospital patients are a bit anemic. And so it was really a challenge of the system. And so we're comparing ourselves to a Mayo Clinic study, an a posteriori method. So they exclude the ICD-9s associated with those conditions on the bottom left. And then the limit algorithm created probably something like 50 codes. Here are the top 10, the first 10 that came out. And then we generated the central 95% from the excluded hemoglobin histogram or population. And here it's comparing the results of limit with both with the blue, which is the currently used interval at the time at Stanford, and the yellow was the Mayo study, a posteriori method, and then red was the automated method. And then you can see looking at this that it performs pretty well. Both the Mayo approach and limit tended to be lower, showing that anemia that you are going to be picking up with the hospitalized patient population. But the automated method performs pretty well. Now this is the challenge of it with hemoglobin. So on the left, you can see if you look at the dotted line, that's the distribution of hemoglobins from the Mayo study before they excluded. And then after they excluded, it's the solid line. So you can see it shifted to the right, which is they're basically excluding a lot of patients with who are anemic, which is good. And using limit, we see a similar thing, right, where you're enriching for this population that's normal. First of all, we end up with something that looks more like a bell curve, which is nice, and it's doing the best it can. I mean, if you look at the reference intervals for hemoglobin in males, adult males, it's up to 17.5 is normal. And there's hardly any patients up there, so it's nearly basically impossible for the reference range to go up to 17.5, which is why in the end we didn't go that high. But I think the algorithm did an excellent job for what you're asking of it. And there's something also to be said, I think, instead of calling everyone anemic in the hospital, to say, well, here's our reference range for hemoglobin for our hospitalized patients. And if your patient is in this range, that's pretty normal. If they're below it, then they're starting to become an outlier. Now we did it, of course, for some other analytes, sodium and potassium. And here we're comparing the red, which is the limit interval, with some other internationally accepted intervals. And you can see that there's a pretty reasonable correlation between them. And one thing you can do with this, which I think could be pretty interesting, is you can reverse it. So what we did originally was we asked, given an outlying hemoglobin value, or not like outlying test value, which ICD-9s are associated with it, we can flip it around now that we have this data and say, well, given these ICD-9 codes, which tests are likely to be abnormal? And that can be the basis of a decision support tool, ordering tool. All right, so now I want to talk about encoded data in routine laboratory panels. And so obviously, in the era of personalized and precision medicine, there is enormous amounts of data, right? And integrating, synthesizing all that data in one, cognitively is impossible. This is a slide from Hemolysis. He's a director of pathology informatics here at University of Michigan. And down on the bottom left is a single analyte, one data element. That's something a provider can look at and make inferences from. And then you get the basic metabolic panel, the Chem7. Now there's 7 elements, and you can glean out of that certain relationships. Like you can look at the electrolytes and calculate an anion gap. You know that the BUN and creatinine are going to be related to help you determine if the pre-renal or renal failure. But there's probably other relationships you may be missing. And certainly after that, when you get to the comprehensive panel and the CBC and on, it's far too complicated for pattern recognition cognitively. And at some point, you're at the supercomputing threshold. And so I wanted to show you this. This is I produced a correlation matrix. So this is looking at the comprehensive metabolic panel. And it's all the analytes in the panel are both in rows and columns. And then if there's a strong correlation, positive correlation, you see a blue dot, and if it's negative, you see a red one. So of course the diagonal is all positive blue because that's comparing one analyte with itself. And then you can see that, for instance, the top total protein is correlated pretty well with globulins, which makes sense. That creatinine and BUN is correlated pretty strongly, ALT and AST very strongly. Sodium chloride correlated for charge balance. But then after that, there's not a lot of correlation. And it's actually good because you want to have a lot of independent measurements in your comprehensive metabolic panel. You don't want them telling you the same thing. But what this won't tell you is if there's a small subgroup where there's a relationship, a correlation between these values that would help you identify and diagnose them, you wouldn't necessarily see it here if it's not, you don't see that relationship with everyone. And so that's what Hemolysis and his colleagues attempted to do for thiopurine metabolite imputation. So this idea of encoded data is that there are these hidden relationships that you might not pick up just by looking at the results with your eyes. So inflammatory bowel disease affects at least 1.6 million individuals in the U.S. And severe refractory disease is typically treated with biologics, which are very expensive, or purine analogs, which are less expensive, but they're very difficult to manage, both for clinical response and toxicity. And the best approach right now is to follow with the thiopurine metabolite, 6-TGN, 6-MMP. But the diagnostic performance of those are very questionable, and they're still pretty pricey. So there's a strong motivation to develop an improved test for clinical management of the IBD patients. So they went to machine learning algorithms. There are several to choose from, and they come in a couple of classifications, but it's basically unsupervised and supervised. And unsupervised, you don't provide any labels on your patient data, who has disease, who doesn't. You just let the algorithms cluster your data into groups. And then there's supervised approaches in which you do label your patients, those with clinical response with IBD, patients with IBD with a clinical response to the purine analogs or no response. And then you create a model that, based on some clinical data, like the CBC and the comprehensive metabolic panel, and try to detect patterns to classify those two groups properly. So they chose a random forest algorithm, which is a commonly used algorithm. And the idea here is that you start with a decision tree, a single decision tree. And you train on a single decision tree, and that is to say, at the first node, maybe there's a sodium. If the sodium is less than 130, you go to the left. If it's greater than 130, you go to the right. And then at the next step, maybe you look at potassium. If it's greater than 5.5, you go to the left, and less than you go to the right, or so forth. And you can go back to sodium after that, or incorporate any number of analytes. But then you do this on a multitude of trees, and then essentially let them vote to classify the next patient after you train the data. And this is the prediction stage. So it turns out the algorithm works very well. So they called it ThioMon, which is thiopurine metabolite monitoring. And you can see on the left, first of all, there's a ROT curve, right? And so that gives you a sense of how accurate your tool is. Now, 6-TGN is the current tool for predicting clinical response to purine analogs. And you can see here, it performs actually quite poorly with the area under the curve of 0.6. So if you're at 0.5, you're talking about a coin flip. There's no information there. So it's better than that, but not much. And then ThioMon is performing with the area under the curve of 80, 85 about, which is much better. And that's statistically better too. And the other thing you can get out of these models, these are all the different analytes on the right that were included in the random forest, and the random forest will tell you which predictors are most important. As you can see, well, it's neutrophils, so something to do with inflammation, alk-phos, RDW, something to do with red cell growth, age, white counts, and so you get some sense of what the algorithm is doing. But what's really powerful about the random forest models is that it automatically and implicitly looks for interactions between the terms, and that's really what makes it powerful. So, making the model is one thing, but actually integrating it into clinical care is entirely another one, and they've successfully done this. So, they're doing a machine learning modeling of the CBC in the Comprehensive Metabolic Panel. Now, the first thing they do is they integrate the laboratory information system orders with routine patient testing. So what that means is that the CBC and the comps are constantly monitored for new paired events, and that's stored in a database. Then, if a ThioMon is ordered, it triggers this computational assay on these paired events. The analysis occurs on a laboratory information system attached to our server, and then that's where the random forest algorithm generates a computational report, which is returned to both the LAS and a web services layer, so that a provider can get a result, a pretty result like this. So you can see here on the right, this is the result, and let's point this out. So in the middle here, you see that there's a prediction of the clinical response. So in this case, the probability of clinical response is 31%. The probability of shunting to toxic NMP is 56%. And probability of noncompliance is 64%. So you get this information out of it. And it's been in use now for several years. And now he's exploring the use of data from generated other institutions. So you take the CBC and comp coming from another institution that uses slightly different reference ranges, normalize those reference ranges, and then still use the same algorithm. Okay, so another thing I'd like to talk about is point-of-care instrument accuracy. So being the director of point-of-care testing here, something that comes up all the time is quality. So there is this great survey that CAP conducted of physicians, not pathologists, but physicians, about the importance of the different laboratory services. And by far, they voted the most important being quality and reliability of results. That's what they need from us. So and quality is particularly important for point-of-care testing, since the central lab is always an option. You can use it. And so yes, you get some benefit of reduced turnaround times, immediate management changes, and so forth in point-of-care testing. But if you can't trust the results, then sometimes this is not worth it. So meaningful use. So the FDA, back in 2007, the FDA Amendments Act directed the FDA to leverage existing information technology to develop more active post-market surveillance for device quality. So basically, there's quite a bit, there are huge hurdles to get a device through the FDA currently. But then after that, the post-market surveillance is kind of thin. There's the MAUDE event reporting database. So if you think that there is an adverse event caused by a diagnostic test, you could submit a complaint. But there's no active surveillance. So I want to talk about how we can accomplish this through mining the electronic health records. So we actually use quite a bit of point-of-care testing at the University of Michigan. On the bottom there, you can see all the different devices we have, and we conduct, there's about 1 million tests performed per year. And so, some of these instruments are being used constantly, right? And at the same time, people are ordering central lab tests. And when those two match up in time, someone orders both a central lab glucose and performs a point-of-care glucose. Well, that's an opportunity for an accuracy study. So instead of throwing that data away, why not look at it? So we call it the algorithm detect, and so the idea is to find these coincident testing events. So along the top, you can see that in this timeline that I'm just depicting that you have several point-of-care tests occurring, for instance, in the ICU, and occasionally there is a central laboratory test. And sometimes they match up, and sometimes they don't match up. And in the case of glucose in the ICU, sometimes the situation might be that there's an unexpected point-of-care glucose result, so then you send it to the central lab. But often, I think more than not, it's sort of random when they match up, or it's easier for a nurse to collect both at the same time. If they're taking it from an arterial line, they can get both samples at the same time. So they match up quite often. And so you identify all these coincident events by mining your laboratory information system. And then we performed a bunch of filters because we wanted to clean the data up as much as possible. So that's down here. So we wanted to limit for the lab turnaround time, because we knew that there might be continued glycolysis for those samples that take an hour or two to get to the central lab for testing, because the red cells are continue to digest the glucose. And we also wanted to limit by location, since some locations did finger sticks, and we thought that would be a confounder. We also wanted to limit to a time period to remove sort of long-term variation. We also limited repeat testing, because we figure in those cases something may have gone wrong that they identified, and maybe there was an IV contamination, for instance. And they also pulled data to know which patients had peripheral lines placed, because then the nurses would be more likely to use the line rather than use a finger stick. And we currently we don't know which ones are explicitly finger sticks or arterial or venous samples. So after filtering, we end up with a data set, and then we can perform an accuracy evaluation. So in a traditional study, you're looking at about, on the left, maybe 200 events, which is a pretty big laboratory study. But using this method, we ended up with about 20,000 events using the minimal amount of filters. A huge number, right? And when you get to orders of magnitude more data points, you can start asking different questions and go to a much more granular level. And so I just wanted to point out some of the effects of the filtering. So on the left, you can see here, these are all the different filters that we were applying sequentially. And then you can see that as we did that, what the impact was on bias. So let's think about bias first. We wanted to get a gold standard. So one gold standard was the laboratory validation study we performed, which was about 200 or so events. And another one is we had nurses in the ICU identify which paired events could be used for the study, because we know that they didn't infuse dextrose or insulin in between the two sample draws. Because we found events that were coincident in time, but maybe they were separated by 5 minutes. And the timestamps aren't perfect anyway. So that was our gold standard. And then you can see here, detect in sort of the burnt orange performed very well, and very similarly both of the other studies, and there really wasn't much of a difference as you added more and more filters. But with the random error, which includes imprecision as well as patient-specific interferences, you can see that detect the imprecision or the random error improved quite a bit as you added more and more filters. And then it matched up eventually with the bedside ICU study. The lab validation study was always a bit better, which you'd expect because there's going to be less variation. 2 instruments were used and 2 people performed the test in the lab who were professionals. And then, on the right side, it gives you, and this is the kind of thing we can start to ask. We have so much data, we can look at bias and random error across the entire range of central laboratory glucose values. So you can start to look and see the extremes. How does the accuracy change? And then here's some other analyses we could run. So in the top left, in panel A, what that's looking at is the time it took to get from the bedside to the lab, central lab. Because we think during that time period there's probably going to be some glycolysis. And on the y-axis this is basically a bland Altman. The y-axis is the point-of-care result minus the central lab result. So it's the error of the point-of-care test. But actually, in this case, it's the error of the central lab, since it's the central lab that's experiencing the glycolysis. So you can see here that over a 2-hour period, there's a shift of about 10 mg per deciliter. So that's 5 mg per deciliter per hour, which is consistent with traditional estimates of glycolysis over time. So then we subtracted that effect out, actually, and then the bias more or less disappeared on the point-of-care instrument. And panel B here, we're looking at the effect of hematocrit. And hematocrit is a well-known interferent with point-of-care glucose meters. You can see here that as there's less hematocrit, then the bias is increasing. The point-of-care test tends to run high. And then we had enough data in panel C to look at that exact same effect, but do it by glucose value. And there was a sense in the literature that this effect was worse as you got higher in glucose values, and in fact we see that bias goes up to 5 to 10% at high levels of glucose, and it's down around 0 otherwise. So getting this huge amount of data allows a fine-grained characterization of accuracy. And we could ask other things, like we regressed, it's kind of a washed out image here, but we regressed the errors of the point-of-care instrument against a bunch of physiological parameters as codified in different laboratory analytes. And so what we did was we looked at what is the range of a given analyte you might expect among patients, and if you were to shift that much, how much of a bias would you see in your point-of-care meter? And so that's what we're seeing here at the top. You see stuff like analytes like hemoglobin, which we know is a problem, causing about 10 to 15% shift in the worst cases. And others like oxygenation, actually from blood gas instrumentation, which may interfere with redox reactions in the instrument. So these are the types of studies you can perform. And one they haven't done yet is looking at the impact and interference of medication. Because of course, that's one of the main concerns in ICUs, is that medications could interfere with this testing. And this is basically the green button for point-of-care quality. It's our ongoing, continuous quality management. And you can basically automate this to pull these paired events for all of your instruments. And glucose is the most data because it's probably half of our testing. But you see plenty of data for other instruments as well. We pulled it for our point-of-care A1c, and this is one where we're interested in it because this recent FDA submission of a point-of-care instrument for A1c to be a WAVE format, but WAVE for diagnosis, which is a step up from monitoring. And so we were looking at what our accuracy was. And there's a bit more variation there than we were expecting to see. So that's something that was a flag for us that we're going to look into. And then, just to give you a sense of how useful this is, we were recently asked, and this happens all the time, provider or nurse contacts us and says, I think your instrument's not performing well. You know, several results in a row were higher than the central lab because I was confirming them. And so that actually they called from the blue, the XCVB, and so they were seeing a positive bias. So we plotted all this data, and you know, it didn't really seem like there was a positive bias overall, but in that one group there was. And so, and by the way, with this method, you have enough data that you can stratify and color code by location or potentially by instrument to look for, you know, the sick instruments. So anyway, we separated out two groups that turned out that one group was using one instrument but submitting it to the wrong code, and then the group that was contacted us was actually submitting to the right code, and we see here, in fact, there is a positive bias. And so now we've got something, a foundation for a conversation to have, rather than just a he says, she says. And one last thing I think I want to say about this, confounds and biases are a really big concern when you're doing these retrospective, you know, studies, data mining studies. And so there's a couple of key. Quadis is a tool to assess diagnostic accuracy studies, and it's really useful for someone interested in laboratory diagnostics and understanding and good study design. But I just want to point out three of these. So number one, what was the spectrum of patients representative of the patients who received the test in practice? You don't want to have a selection bias choosing the wrong set of patients. And that's a possibility. So with glucose, when there's sort of a random pairing of the point of care with the central lab testing, it doesn't matter. It's random, so it can happen to any patient. But in a case where you don't have that much testing, or you don't have a system set up like we do with troponin in the ED, where there is an automated confirmation of high values, well, then you're probably only going to get confirmation, or possibly only confirmation, when there's an unexpected result, which is more likely to be wrong, and you might be getting the worst case scenario. Though there are still events that are occurring, you know, regarding the accuracy study. So another thing is, another one I thought was important to highlight is number 5. Did the whole sample or just a random selection of the sample receive verification using a reference standard of diagnosis? So this is really important when you're trying to develop a model, a machine learning model to predict something, for instance. So if you want to look at the accuracy or trying to predict celiac disease, and you're starting with the antibodies, the anti-tissue transglutaminase, for instance, you want to make a model predicting positive biopsy, and of course using the positive biopsy as your gold standard. And you can look retrospectively and collect all that data. But the problem is that not every patient is going to get a biopsy, right? First of all, you're more likely to get a biopsy if your anti-TTG is high. And then you're more likely to get a biopsy if it's low, but you have other risk factors. So you have an implicit selection bias there that you need to be really careful about when doing these types of studies. And then the last one I want to mention was the reference standard independent of the index test. So did the index test form part of the reference? Now it sounds kind of ridiculous, but in point-of-care testing this happens all the time, where someone's doing a study with multiple point-of-care instruments, and then it's sort of a winner takes all. So the majority vote wins. So if most of the instruments vote one way, then that's considered positive. But that, of course, makes it tends to enhance the accuracy measurements of your test of interest. So be concerned, on the lookout for that. Now, another one I wanted to talk about is hemolysis, and this is a big problem. It happens a lot, more in some from some units than others, and there's a lot of miscommunication about this, and you can hear from someone out on the floors, well, why are you hemolyzing all my specimens? Because they get canceled a lot and so forth. Of course, we're not hemolyzing them. They get hemolyzed in the pre-analytic phase when you're drawing the blood. But hemolysis can interfere with a number of laboratory tests. So you could simply get a release of an analyte that's of high concentration in the cell, in the red cell, like potassium, or there can be interference with some of the enzymes in in a reaction like a bilirubin, dilution like glucose, or release of a thromboplastic substance that interferes with coag testing. Proteolysis could occur, which could interfere with protein testing, like for insulin. And so we wanted to look at mining the LAS for the impact of hemolysis. And one of the reasons is that we actually currently report all of our potassiums, regardless of hemolysis, but we report what level of hemolysis there is. And we'd like to switch over to canceling some results if there's a high enough level of hemolysis. So we needed to decide what level of hemolysis is high enough to warrant canceling the test. So we decided to go through. So we identified potassium results first that have hemolysis, and we assigned a hemolysis index to each one. Now, ideally, you would simply have the hemolysis index in the medical record or the LAS, but we don't, and this is the kind of thing you run into all the time trying to mine through the health record. So instead, you get this really crazy statement that's hardly legible, but it's constant, it's consistent, and so you can query for that within R to find all those events. Then we identify, again, near in time, non-hemolyzed potassium results. Maybe they sent in a sample right after because it was hemolyzed and they didn't trust it, and that happens a lot. So we want to use those pairs to see what level of hemolysis is causing elevation of potassium that's concerning. So we went ahead and did this, found about 6,700 hemolyzed events and about over 500,000 non-hemolyzed events. And it looks something like this. So this is looking at samples collected within 60 minutes of each other. And the H index is our laboratory system's method of assessing how much hemolysis. So 2, 3, and 4 are increased amounts of hemolysis. And number 4 is marked hemolysis. And so you can see on average we're getting a shift of about 3 with potassium when there's marked hemolysis. But we can do a little better than that even and create essentially what is a lookup table. So on the x-axis here is the index potassium result you're concerned about. On the y-axis is the near in time unhemolyzed event. And you can see here, and we color-coded by hemolysis. So the gray ones, there's no hemolysis. And you can see there, you get a nice correlation curve of a slope of about 1, which is what you'd expect. There's a little bit of variation there, which is imprecision, implicit, and instrumentation, but probably the fact that 60 minutes had elapsed, perhaps. But then as you increase hemolysis, you see, like for instance, those in red, it's just flat. So with marked hemolysis, even if you have a potassium of 8 or 10, there were no events that were above the critical value of 6, except one. And you're always going to see this with messy data. But that particular patient, I looked and it appeared as though the unhemolyzed paired event was actually hemolyzed. It just wasn't recorded properly because there was a blood gas performed at the same time that showed a normal potassium. And then this red one down here was an IV dilution. That's why the potassium was low. So with this data now, we can have informed discussion about which tests, what level of hemolysis we should not report, and have it be a data-driven decision. So the last thing I wanted to talk about is modeling, and I want to talk about it because you can pull wonderful information and data out of the health system to inform your health system and improve it, but you really need decision models to make the most of that data. This is how you synthesize the data into some conclusions. And when you're making models, it's really important not just to look, if you're looking at diagnostics, don't just look at sensitivity and specificity. If you want to know how a diagnostic is going to impact a patient outcome, you not only have to look at the issues, the functions in green, which have to do with the test and the accuracy within the laboratory and the time of turnaround. You also need to look at how those tests are going to impact the diagnosis. And if a diagnosis is impacted, that's not quite enough. You also need a management change, and then you need the drug to work. So there's a lot of steps that have to be successfully completed before a diagnostic test will have a positive impact on the health system. Now here's I want to show you guys two models, one a very simple one and then a more complicated one. So this is a study by Brad Karen and his colleagues. I'm using their method. This is our data. This is a histogram of glucose values from the ICUs, and they're using a glycemic control protocol with these decision limits. And so what this means is these are all decision limits where a different amount of insulin would be administered. Now, this is assuming that all the results are perfectly accurate. Well, what if you add on bias and imprecision to any of these results? Then they might shift. They might cross over into a different they might cross over a decision limit and cause the inappropriate amount of insulin to be delivered. So what they did was they plotted the bias on the y-axis and imprecision on the x-axis, and then plotted the lines showing the rate, for a given imprecision and given bias, what percent of cases we misclassified by 3 categories, not 1 or 2, but over 3 different categories. And so when you do that and place your meter, ours is occurring somewhere there, and it's consistent with what I've been finding with glucose meters, which is that they're pretty accurate. Now, if you look at the actual, instead of basing this on estimates of bias and imprecision, just look at the actual paired events that we had to see how many point-of-care tests missed hypoglycemia, for instance. Well, there's 364 hypoglycemic events in the central lab. A third of those were missed by the point-of-care instrument, but they were just over the edge a bit, and in the end, only 2 of them would have been given insulin that weren't, or only 2 would have been given insulin inappropriately. So the problem is pretty minimal. Now you can get to more sophisticated models by looking at cost effectiveness. And so this is the cost effectiveness plane or space where you have cost and effectiveness on different axes. Sometimes they're flipped. And of course, you want to have great effectiveness and low cost, but you plot all of your different scenarios or strategies on the plane, and then can make a decision about which one you want to use. But you can use this to ask questions like, how much should we be willing to pay for a more rapid turnaround in C. diff testing, or how predictive of a positive biopsy does anti-TTG screening need to be in celiac disease? So there are many approaches to this, but 2 common approaches are decision trees and microsimulation. So a decision tree is simpler. It's a lot like the random forest, right? You start off with C. diff testing, and then your 2 strategies on the left, the on-demand PCR is one strategy, and ELISA is another strategy. And then from that point, there's a certain chance that the patient will either have C. diff infection or not, and a certain chance that the test will be positive or not, leading to the final terminal nodes of true positives, false negatives, true negatives, and false positives. And you put a cost on those events, and then you know what percent of the population falls in each category, and then you can roll the model back and get sort of what your average cost and effectiveness is going to be. A microsimulation, so I'm showing you data on an HIV disease progression model I've been building. So this focuses on sub-Saharan Africa, but I wanted to mention this in the context of big data, not only because it's useful to synthesize big data, but it is big data itself. So some of the heaviest computational needs I have is to run this model. So you start with an initial cohort, and that's shown on the left here. And we have healthy patients, HIV undiagnosed, HIV drug naive, HIV on heart 1 or second line heart, or those who failed heart. And so that's your initial population, and then you model this month by month over like 10 to 30 years, and all along you're tracking things like there's a testing event maybe, or a medication change, and you're tracking the viral load and how the viral load is impacting the CD4 count. And a low CD4 count predisposed to an opportunistic infection or to a death. And so doing this is easy to say, all right, well, we're going to add some error into the test, or we're going to change the algorithm. It's supposed to be using two different brands in a parallel testing approach, but maybe they're just using one brand because the other one was out of stock. And so what is the impact that those kinds of decisions have on accuracy, misdiagnosis of HIV, and then outcomes and costs? Now, on top of that, you want to get a sense of uncertainty. And so what I've included is a probabilistic sensitivity analysis. So instead of a parameter, like the cost of a rapid diagnostic test for HIV, there's a distribution, like a bell curve. And so then you need to run the simulation many times in order to get some confidence intervals. And this is done in parallel processing on multiple cores. And so when you do that, you see on the right here, you can track your different your cohorts. And so you see right away, this is looking at the test and treat strategy, where you're immediately treating anyone regardless of CD4 count. And so immediately a lot of people go on to heart, the prior diagnosed patients, and that continues to go up, and it starts to drop eventually, and those going on heart on second line is starting to increase. You can see those that are drug naive is decreasing. And then you synthesize all that to look at your question of interest. So in my case, I was looking at the impact of testing frequency on false positive diagnoses. So if you diagnose if you're testing over and over again, the high frequency annually, and you've got your specificity is a touch low, well, you're going to end up misdiagnosing a lot of patients. And so the literature is actually shows some evidence, disturbing evidence of misdiagnosis of HIV. And so this is looking at the impact of testing frequency, because you also need to increase testing frequency if you want universal coverage to get patients when they're newly infected. And then finally, and you can take that data and put it onto the cost effectiveness plane like this. So costs on the X and Y axis is the quality adjusted life years. And green is high quality testing, and orange is the current or low quality. And so with this data, you can get a sense of how much it's costing the health system to have the sort of current low quality testing, and also how many adjusted life years are you going to gain by going to high quality. And that gives decision makers a sense of how much they can invest in quality improvement. All right, well, that's what I wanted to share with you. I wanted to acknowledge some people. So with reference interval generation, Sarah Poole and Nigam Shah at Stanford. Machine learning and encoded data, that's Yuval Belisa's work with his colleagues in pathinformatics here. Pre-analytic error and hemolysis is David Manthei's resident in pathology here, has been working on that with me. And on the point of care accuracy stuff, Nick Meisner, Denise Rakowski, David Manthei, and Don Chicario, all in pathology, and Nigam Shah at Stanford. And I thank you for your time.
Thank you, Doctor, for that informative presentation. And before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We're going to try to answer as many questions as time allows. Doctor, our first question is, why is big data in healthcare taking so long?
Thank you. Yes, it is taking a long time. I think there's a few key reasons for this. I think, of course, part of it is incentives. So with the increasing pay for performance and capitated models, there's going to be a strong desire among hospital administration to prevent illness and high cost care, and using the learning healthcare system and big data from electronic health records are going to be a big part of that. So I think this is it's unavoidable that this will continue to develop. Another big problem is the data is stuck in the medical record. It's there. How do you get it out? And I think maybe one of the problems is that there's not a ton of competition between electronic health records, and there's not a strong incentive to put this analytics at the touch of a finger. I mean, eventually it might get there. So now groups are doing it outside of the electronic health record itself, but that means having to invest in analysts and so forth, which hospitals are doing. This is a massive industry, right? It's 20% of our GDP in the U.S. and there's a huge amount of waste, right? Don Berwick published on this models of or Wedge's model of inefficiencies, 6 categories of inefficiencies that account for 20% or probably more of healthcare dollars. So I think it's just a matter of time.
Thank you. Our next question. Do you really need to use all the filtering steps for the POC accurate studies?
Right. That was a complicated study that we performed. And it was complicated because it pulled data from more than one place, right? So we had to get arterial line data from the medical record, the electronic EMR versus most of the rest of it came from the laboratory information system. But I think the answer to that is you probably don't. So, timestamps are funny things. You see a timestamp in the medical record and you think, oh, okay, that's exactly when this glucose was taken. Well, that's not the case. So the point of care, that is, the time is when you submit the result. That's pretty close. But for the central lab, it's the time you print out the tube label. And so if you take a while to get over to the patient side, it's going to look like you drew it earlier than you actually did. So we did a study to actually identify the delay in time between the label printing and drawing on average, and we used that adjustment. But in the end, it didn't help at all to do that, and actually made things a little bit worse. So I think you can just base it on time 0, assume there's no errors, and then choose your time window. The delays to the central lab were a big part of the bias. In fact, it accounted for all the bias you saw in the point of care instrument. So be careful with the bias, and you might want to correct for that. But you could also just know that that's probably causing about 5 mg per deciliter if on average you're taking about an hour to turn around your glucose in the central lab. The arterial lines made very little difference. And we did that, of course, because if a result came from a finger stick and you're comparing to an arterial line sample or venous line sample, that's a confound. They're not really comparing apples to apples. And the A-lines made little difference, I think, because location pretty much accounted for all that. Once we limited it to the ICUs, they have a lot of A-lines and a lot of lines. So there's very little finger sticks that are occurring there. And limiting to location is quite easy. And then the other thing I want to mention about this is just the bias that you might get when there's not a built-in system for confirmation. So, to be concerned about results, central lab results being ordered simply because there's an unexpected point of care result, which could select possibly for a worse result. Thank you.
And our next question, how are we supposed to do this? I don't know R, and I don't have access to data from the EMR.
Right. Yeah, so if you're interested in this, you can make progress. I think the first question you have to ask is which data do you need? So is the data going to be an electronic medical record or in the laboratory information system? If you're in pathology and clinical diagnostics, then you're going to have easy access to the laboratory information system. And what's beautiful about that is all the data is formatted. And that's really special because it's easy to work with. The medical record, there's some formatted fields like ICD-9 codes, but most of it is free text. And then you got to start worrying about natural language processing or annotating the medical record. Of course, doing it by hand is taking a deep dive into the medical record and reading, doing chart reviews. So anyway, that's an important first step. But some things, the laboratory information system is very limited. You have formatted data and objective data, but you don't have information typically like the ICD-9 codes for the patient, and that stuff is gold. So you're going to have a contact at both. And most institutions, I think, have an analytics service that will, sometimes for a fee, collect the data for you and give you data down. So if you're proficient in the computational methods, you just need to get IRB approval and get that data down. And if you're not, from my experience here, analysts are one of the fastest growing positions in the hospital system. Hospitals know that they need analysts. And so you just need to find where they are. Are they, we have a strong path informatics group. We also have a quality division, and there's analysts in both. That's just within pathology. And then, the hospital has the clinical informatics group, where there's more analysts. And there's a lot of people asking them for help, but if you have a compelling question, I think you can get someone to help you succeed. And, I think try in our class. They're all over the place. You can see them on Coursera and other, the Hopkins has some really good R classes that are free to take. And, if you do this and you've got, a computational sort of skill set, it'll really help you spread your wings and explore, be able to ask your questions, in this wild west of the healthcare system.
Thank you. I'm very intrigued by the ThioMon work. What are the biggest obstacles to making it happen?
So, the biggest problem facing the creation of the machine learning models is phenotyping. So I was creating a model for celiac disease, and it required me to sift through hundreds and hundreds of medical records, and it took an enormous amount of time. And so that really limits how quickly and for how many conditions you could create models. And I think that's one of the greatest problems to be solved in this informatics field, which is the automated phenotyping. ICD-9 coding is known to be imperfect. Can you automate phenotyping with electronic health record data so that cohorts could be automatically generated? And then you could just run machine learning algorithms on any clinical data you happen to have access to on these phenotypes and create models constantly. And sometimes the machine learning part of it is the easy part. And once you have your data in the right format, you can use R to run any number of machine learning algorithms with a different selection of a function. But you can also find plenty of analysts or engineers who are interested in this, who want to have access to data and want to help out. So I think that the key thing is the clinical question and curating a clean gold standard. And that's the key. And then, of course, you're going to need to interface the LIS to an R server. And so it's going to require some informatics expertise.
All right, thank you. And we do have time for one last question. Can you really trust models? Aren't they so dependent on your assumptions that you can lead to nearly any conclusion you want?
So, some ways, yes. And so I think that the bottom line is that you shouldn't take just the headline of a modeling paper. I think that models are completely dependent on the assumptions. That's true, and the real value of modeling is achieved when you're working closely with the decision maker and sort of iteratively adjusting the model to look at different scenarios, performing sensitivity analyses to say, well, if we do this a little different, how will that impact things? And I think it is very dangerous just to read the headline on a modeling paper and then assume that that is fact, or that's how, if you implemented their strategy in your health system, you experience the same outcome. So it's really a decision support tool and not fact. One of my decision analysis professors, Doug Owens, would say that all decisions you make are made with uncertainty. And the advantage of modeling is that you're explicitly stating that uncertainty and where that uncertainty is found, and you can adjust that uncertainty. So how couldn't that be a benefit? Thank you for the question.
Thank you, Doctor, for answering all of the questions here that we have time for. Do you have any final comments for today?
I urge those in the healthcare system that are interested in this kind of work and taking advantage of all that data that's sitting in the health record. Just every time you come up with a problem you're facing, just think, well, could we solve this through a data mining approach? And most of the time, maybe the answer is no, but occasionally you run into a yes. And as you identify those projects, you'll have an arsenal, and then at some point it's worth it, right? To go through the process of learning how to get access to the data and finding people who are going to collaborate with you to analyze it if you don't want to analyze it yourself. But it's just a really exciting world to be involved with and try to take advantage of. So I wish you the best.
Thank you again, Doctor, for your outstanding presentation and for your answers to today's questions. I'd also like to thank LabRoots for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through April 2017. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to any of your colleagues who may have missed today's live event. Until next time, goodbye.